Hello, my name is Uzziah Urquiza, and I am from Southwestern Oklahoma State University, and I will be presenting research conducted over the preliminary exploration of the molecular mechanism of the anti-cancer activity of a novel lead lactate dehydrogenase inhibitor against pancreatic cancer. Pancreatic cancer is one of the leading causes of cancer-related deaths in the United States, and its long-term survival rate is very, very low. It lands just under 10%. Currently, there's not many options for treatment, with chemotherapy being the standard treatment. However, a rapid development of resistance to these chemotherapeutic agents has created a bottleneck in survival rates. This has made it very important for us to find a new biological drug target, and exploring the mechanisms which lead to this resistance can reveal potential therapy targets. The target we are investigating is lactate dehydrogenase A, which is a good target for various reasons. Firstly, being that it is the primary metabolic enzyme that facilitates the process of aerobic glycolysis, which converts pyruvate to lactate in the presence of oxygen. Cancer cells prefer this as it gives them a pathway to many different processes, and the overexpression of LDHA is common in many cancers as it plays a role in several different metabolic pathways as seen in Figure 1. Thirdly, the inhibition of LDHA is not lethal to the cells, so altering its activity should not have a detrimental effect on the cells. The LDHA inhibitor we are studying will be referred to as compound 23. The objective of the study is to understand the molecular mechanism of the anti-cancer activity of compound 23 and to determine if compound 23 is in fact promoting apoptosis. The cells studied are of the Miapaca-2 cell line, a pancreatic cancer cell line, and these were cultured in Dulbecco's modified Eagles medium and then supplemented with 10% fetal bovine serum and 2.5% horse serum, which was incubated at 37 degrees Celsius in a 5% CO2 atmosphere. After culturing, the cells were seeded in 6 well plates and left overnight. The following day, we replaced that medium with serum-free medium to prevent contamination from the proteins commonly found in serums. We then treated those cells with 50 micromole concentration of compound 23 for a full 24 hours. Protein extraction was completed using around 150 microliter ice-cold radioimmunoprecipitation assay buffer, or RIPA buffer, supplemented with protease inhibitors to prevent the degradation of our protein samples. Using gentle scraping, we aided the lysing of the cells by causing mechanical damage. Treatment with RIPA lasted 24 hours, and then cells were aspirated and washed with PBS. The cells were then collected in an Eppendorf tube and incubated in ice for 15 minutes. The protein was quantified using the bisynchronic acid method. A calibration curve was made in order to calculate the protein concentration in unknown samples. The detection of our proteins was done using Western blotting. 35 micrograms of each protein was supplemented to bistrous gels, 4 to 12%, a polyacrylamide gel optimized for protein separation, especially under degradation conditions. The PVF membrane was incubated with the corresponding antibodies for each protein. The results obtained revealed inhibition of LDHA enzymatic activity without inhibition of its expression. As seen in Figure 4, DMSO was used as a control and actin was used as the loading control. There was little effect on the BAX and uncleaved PAR proteins, but the Western blot analysis did reveal activation of pro-apoptotic proteins like the cleaved PARP. It also indicated a decrease in expression of anti-apoptotic proteins such as uncleaved caspase-9 and B-cell lymphoma 2 proteins. These results can lead us to the conclusion that, at least initially, our findings suggest an anti-proliferative effect of compound 23 in these pancreatic cancer cells due to the inhibition of anti-apoptotic and activation of pro-apoptotic proteins. Below our conclusion, you can find our 5 references used in this poster, and then we would like to go ahead and acknowledge and thank the Guy Hagen Endowment Fund and the College of Arts and Science grant for their continued support. Thank you all for listening, and I believe that is all I have.